Good morning, everyone, and welcome to our UNC Health Game Changer series. My name is Leslie Nelson Bernier, and I have the privilege of serving as the president of the UNC Health Foundation. And you are in for a real treat today as we start a discussion on chimeric antigen T cell and the work that we're doing with that program. We call it CAR T for short. And so you'll get to hear a whole lot more today from Dr. John Serody. I'd like to encourage you to please ask questions of our speakers. Your question and answer feature is available for use during this entire session. So please pop your questions in there if you have things that you'd like to talk about with our speakers. And to kick us off, I would like to ask our CEO of UNC Health and Dean of the School of Medicine, Dr. Wesley Burks, to bring some remarks. Dr. Burks?
So Leslie, thank you. And thank all of you for being on today. I'm excited to be joined by Dr. John Serody, as Leslie just mentioned. John is our Elizabeth Thomas Professor of Medicine, Microbiology, and Immunology. Among the many roles, he's the Associate Director for Translational Science at UNC Lineberger. He is the Chief of the Division of Hematology in our Department of Medicine. And at UNC Health, he's the director of our cellular therapy program. And I'm grateful for John and his really busy schedule that he's taken time out to talk to us today about work he's doing and answer your questions about the work that they all are doing to change people's lives. For the Game Changers series, we're changing to move away from just talking about COVID that we have for the last several months, but unable to leave that totally because of where we are. But from a system standpoint, we have about the number of patients that we did in April of 2020, which is pretty amazing. Most of the facilities across our system, and there are 14 of them now, have less than 10 in the hospital and a fewer number in the ICUs, which is really good for the people that live in these regions. From a vaccine standpoint, we've given more than 400,000 vaccines since mid-December. The program itself is transitioning into a new phase to give the vaccines in our ambulatory care and primary care clinics. The big vaccination center here in Chapel Hill at the Friday Center, which has been an unbelievable place to work and give people their vaccinations for the last 6 months, will be decommissioned in late July. We're planning a big event to thank the people that have worked there, like tremendous work that's been going on there for the last 6 months. So now we'll transition back. And again, John, thank you for being with us today. If you can start by telling us a little bit about your history with the work that you're doing and how you came to this research in immunotherapy.
And I want to thank all of you for spending time with me today and taking time out of your busy schedules to participate in this, and thank Wesley and Leslie for inviting me. I've been interested in immunology since probably grade school, so I've always been interested in immunology, and I started off that interest as a stem cell transplanter, which was the first form of immunotherapy. And kind of my goal in developing this program came from my time out in Seattle when I was a transplant fellow and an oncology fellow. And I got the privilege of being on the pediatric ward at the Fred Hutchinson Cancer Research Center and spent 3 months there and got to become really close to a 6-year-old child who come from New Jersey to be treated for what's called juvenile CML, which is unfortunately a much harder disease to treat than adult CML and typically requires a stem cell transplant. And he had multiple complications from his transplant, was in the hospital for 80 days. And I was the primary person who took care of him for most of those 80 days. And unfortunately, he eventually passed away. And what really struck me at that time was kind of 3 things. First was how different he looked from the child that grew up in New Jersey. So the child I got to know from all of the things that we had done to him was a much different child than the one who grew up playing in New Jersey. The second was his mother asked me to perform the eulogy at his funeral. And I was pretty surprised and stunned by that request. And she said, well, you know, you're the only person we know in Seattle. We don't really know anybody. All of our families back in New Jersey, 3,000 miles away, they couldn't come out here. Would you please do this? And I did that. And then the third thing is I told her, and then when I came back here, told the folks here that I wanted to develop a program that didn't require you to go 3,000 miles away to get cutting edge, state-of-the-art therapy, that we should be able to do that in North Carolina, regionally, locally for the individuals in this state. You shouldn't have to do what they had to do and be by yourself in the worst part of your treatment. And so that was the impetus for the development of this program, which started in 2007 with discussions with Shelley Earp and then Ned Sharpless, and then a commitment by Ned when he was the Cancer Center director to develop kind of a state-of-the-art immunotherapy program.
So, John, it's amazing for all of us how we got to where we did in the careers of not only the clinical work, but the research that everyone's doing. Everybody has truly a unique story. So if you would tell us a little bit more about CAR T and how does it work? What's the acronym mean, and
Sure.
How do we understand it better?
And I'm going to share my screen and we'll start.
Okay.
So hopefully you guys can see my screen.
It's good, yes.
Okay. So I wanted to kind of just give a little bit of background and some notions of kind of where we were and where we are now, because I think we've had a revolution in the last few years around the use of the immune system to treat cancer that really has changed what we've done. And really the modern treatment of cancer isn't that old. It's about 130, 135 years old. The development of the radical mastectomy by William Halsted back at Johns Hopkins in the 1880s, and the first use of radiation to treat skin cancer around the early part of the 20th century. Then the first use of antimetabolites by the Sidney Farber at the Dana-Farber to treat leukemia in 1947. And then kind of the whole notion of what I've done for most of my career, which is combination chemotherapy in the 50s, and then the development of small molecule inhibitors to treat specific types of disease in the early part of this century. But the real big kind of change in the way we do things has happened in the last 10 years. And this is on this timeline. And I show the picture here of Emma Whitehead, who was featured in the New York Times. She's a 6-year-old girl who had pediatric acute lymphoblastic leukemia. She had failed all forms of therapy, including an allogeneic stem cell transplant. And the last ditch hope they had to treat her was a novel new therapy called chimeric antigen receptor modified T cells that targeted a protein called CD19 on the surface of her tumor cells. She received this therapy at Penn in 2011 and is alive now, and I think off to school and going through college and the rest of her life. So this was kind of a game changer for those of us who've been in this field. And you see kind of the other highlights that I show here for immune therapy, which really has become the kind of pivotal fourth arm of therapy and is probably the most promising, at least in the last decade. So what is CAR T cells? So it's not what my mother-in-law used to think about when she thought about fast-moving cars. I give you the kind of acronym here. It stands for chimeric antigen receptor modified T cells. And the chimeric antigen receptor is built around the notion that T lymphocytes, which are the killing lymphocytes, are really good killers, but they're lousy at finding targets, where antibodies are really good at finding targets, but lousy at killing. So we've kind of done the less filling, taste great approach to immunology and combined the two. And kind of, I'll show you this. The way we do this is through viral DNA integration. So we take either a lentivirus or a gamma retrovirus, and we generate a virus that encodes for a protein that has parts of the antibody that can stick to parts of an antigen on the tumor cell. We insert that virus into the T cell. We then get the T cell to now express a chimeric receptor, that is a receptor that's got part of the antibody part on it so it can recognize the antigen. It will then find the antigen as shown here. And once it finds the antigen, it starts to multiply, and it multiplies hugely. So you can expand these cells phenomenally, both in the lab and in individuals. And then they elaborate both proteins that can kill tumor cells as well as directly killing tumor cells. And you see in the middle here, a dead tumor cell. And so this approach allows you to now treat any tumor cell where you can find a protein that's specific for the tumor that's not normally found in the body. And so at UNC, we've developed a process for generating CAR T cells out of our manufacturing facility, which is about 4 miles off campus across the highway from the Nantucket Cafe. And this is basically the schema for doing it. We collect blood from an individual. We can do this by apheresing with a machine, or we can just collect blood out of your arm. Usually it's typically 7 to 10 tubes of blood. We take those tubes of blood to the facility. We activate the T cells with antibodies that will make these T cells grow. And then once these T cells start to grow, we insert again the virus into these T cells. These are now CAR-modified T cells that we expand even further in the facility. And once we get them to a certain number that we think are adequate for treating patients, we test them to make sure that they're safe and that they work and they're not contaminated. We freeze a large aliquot of these, and we bring the patient to either the infusion center or the hospital, give them chemotherapy to give them some space for these cells to grow, thaw these cells, and infuse these cells, basically right back into an IV, and then wait for a couple of weeks for these cells to work. And so I'll stop for questions, Wesley, at this point, and then if not, I can move on to kind of what we're doing.
Okay, there are a few questions that people submitted ahead of time that you've talked about, like CAR T therapy itself. But for right now, talk a little bit about the types of diseases that you're really targeting CAR T for. What are the space that you're in mostly?
Yeah, and let me put that a little bit into that, because I have a slide that'll clearly just address that for the future directions and UNC trials. And there's a large number, and we're getting substantially into the solid tumor world. So I want to get into that in a second.
Okay, good. In that part of it. So one of the questions on the chat already has been, are the CAR T cells developed under GLP or GMP conditions?
Yeah, so they are clearly developed under GMP conditions. We have a GMP facility that's a kind of state of the art. If you know the vernacular ISO 7 clean rooms, and it's done all the way a pharmaceutical company would manufacture their drugs. So we do it the exact same way to the exact same exacting requirements of the Food and Drug Administration.
Okay, so the next question is what's going on here at UNC Lineberger? There are also commercial products being developed. How do those kind of all fit together?
Yeah, and I think that's a phenomenal question. When we started our work, which is probably 5 or 6 years ago, as I indicated from Emma's story, the group at Penn had developed a CAR that was specific for this protein CD19 on B cells that could be used to treat children with leukemia. Novartis took that intellectual property and developed that into a drug called chimeric. And so we've, by and large, not focused on the areas where there are commercial CAR T-cell therapies that are available. What we focused on are areas where commercial companies don't have products, or we focused on ways to make the commercial products safer. And I'll show you a little bit of data in a few minutes around a safety switch that we've incorporated into our CARs that limits the complications that these cells have.
Okay. Another question talks about the FDA approval process. And just from a big picture, help people understand how that happens. What happens? What has to happen there?
Yeah, so, and I'll go through this for our lead product, which is a CD30 CAR that's directed against an antigen that's found on the surface of Hodgkin's lymphoma cells. So typically there's three phases of clinical studies that we engage. The first phase is called phase 1. It's usually a safety phase where you basically are looking at whether the treatment is safe and trying to figure out to some extent if it works and what the right dose is. The second phase is then taking the phase 1 dose and doing a much larger study to see how effective your treatment is. And then in the third phase, usually compare your treatment with either standard of care or if there's no standard of care, placebo. Most of the studies that I've engaged at UNC are in the phase 1 to 2 area. And then what we've typically done is handed our products off to commercial companies to try to do FDA registration studies in either an expanded phase 2 or phase 3 setting, which is what Tessa Therapeutics is doing with our lead compound, which is a CD30 treatment.
Okay, and give them an idea about that time frame of what you just talked about. That doesn't happen in a few months.
No, and so not even in a few years. So the initial description of the CD30 CAR was made by a phenomenally talented researcher here, Barbara Savaldo. when she was at Baylor, probably in 2006, 2007. They did an early study of the CAR T cell for CD30 around 2011, 12. Didn't really have a lot of activity. They brought the product here. We refigured it and added some things to it to make it work substantially better. Did those studies in 2015, 16, and 17, published that data. As I said, this company then is opted to develop that intellectual property from us, and they're going to do a large pivotal study starting this year. And we hope that this will be registered in front of the FDA in 2022. So you're looking at a 13 to 14 year timeline from the initial thought and the preclinical work in the lab to getting the drug approved.
So in that phase 3 process that you just described to eventually have the FDA approval, what numbers of patients do you really talking about before that approval will come?
Yeah, I think we've been fortunate. I'll show you a little bit of information that we've been fortunate to get fast track designation by the FDA, I think because of the phenomenal success of this treatment. And so the thought is you may be able to do the phase 3 study with potentially 100 patients, 50 in each arm of the study. That's much different than a typical phase 3 study for a drug that doesn't have fast track approval, where you may have hundreds to thousands of patients in those studies. So I think that's been fortunate for the company, and I think they hope to be able to complete those studies in a fairly time-sensitive manner, maybe in the next year, where if you had to do hundreds to thousands, it probably would take us 3 to 4 years to do those studies at multiple different sites.
And for many people now reading about the COVID vaccination process, I would have seen that for each of the companies that they needed in the range of 30,000+ patients that were in phase 3 to be able to get the vaccine and then get the approval process. So just the differences in the two types of treatments. So John, just a little bit personal on this part. When you wake up in the morning, what excites you about this therapy? What drives you?
What doesn't? I mean, I think that it's probably, to me, been the best time to be in tumor immunology in my career. I mean, we basically, I started doing this work in the late 90s, and we were in the desert, Wesley, for probably the first 10 years, with a lot of folks asking, why do we put money into this type of studies? They don't work. And it's really been unbelievably encouraging to see all the progress we've made in the last 10 years. And, I've treated a number of individuals who failed everything. I mean, stem cell transplant, allogeneic, 2 different stem cell transplants, all forms of therapy, probably had less than 6 months to live, who are now 2, 3, 4 years out from that treatment and doing fine. And so, that's phenomenally rewarding, where you can see something that you've worked so hard for 3 decades come to fruition for patient care.
Yeah, no, it's pretty amazing what you and your colleagues have done and continue to do. So why don't we go ahead and leave this part and go ahead to the next part of your presentation, because many of the other questions are really around the future directions, the types of tumors, the side effects, things like that. So go ahead, please, John.
And then please feel free to ask whatever you wish of me. I'm happy to answer as many questions as I can. So as I said, the initial commercial therapies were around a B-cell antigen called CD19, and were used to treat B-cell non-Hodgkin's lymphoma and acute lymphoblastic leukemia. Barbara Sabaldo, initially when she was at Baylor, and then when she came here with Gian Pietro Dotti and myself, developed a CAR T cell product specific for a protein called CD30 that's on the surface of Reed-Sternberg cells that I show in the upper right. That's for Hodgkin's lymphoma, a different disease. And in the majority of individuals, Hodgkin's lymphoma is curable with chemotherapy, but about 20–30% of the time, and slightly higher in older individuals, initial chemotherapy doesn't work. Those individuals can be salvaged with more chemotherapy and a stem cell transplant, but maybe half of those individuals aren't cured. And it's that group of individuals that has a really, really, unfortunately, poor outcome, with most of those individuals not alive 3 years after relapse from a transplant. So we developed this CAR T cell specific for CD30, and here's the schema that we used for this pivotal study. We procured the cells, as I talked about. Typically, it's just out of the arm of individuals by drawing typically 10 tubes of blood. It takes us about 4 weeks to manufacture the cells. So you could get some bridging therapy to hold you, although, as I said, almost all of these patients had failed every conventional therapy. So most of the bridging therapy was pretty modest. We then give you a lymphodepleting chemotherapy drug. And that typically was initially a drug called bendamustine. And then I think what we really developed that was substantially better was this combination of bendamustine with another chemotherapy drug called fludarabine that was given before the cells. The cells are there, then infused, and then we follow you to see how you do. And this was a trial that we initiated at UNC, and then the folks at Baylor, based on the phenomenal initial success that we had, started their own trial using a combination of two drugs, Cytoxan or cyclophosphamide and fludarabine. And there you see the objectives. Initially, it's a phase 1 study safety, and then we looked at response. And so this is what we found. And again, I'll compare and contrast this, for instance, to checkpoint inhibitors, which are used for the treatment of Hodgkin's lymphoma. Nivolumab was the first that was approved in this same group of patients who had failed stem cell transplant. Checkpoint inhibitors have about a 15% complete response rate. That CR rate is shown here, and about a 50 to 60% overall response rate. You can see for us, and this is now up to, this was the initial publication in the Journal of Clinical Oncology. We've now treated 32 patients, and these numbers still hold. The complete response rate is around 75%. So 5 times what you see with the commercially available therapies, and greater than anything that's ever been published in this group of patients. And you see the overall response rate is 80%. Which for a group of patients that's failed everything is phenomenal. And our response rate with our approach was a little bit better than the response rate that was seen in Baylor, although the Baylor response rate was still quite substantial with a 50% complete response rate and 18% PR rate for an overall response rate of 65%. And you can see here that the addition to fludarabine really makes a difference because the small number of patients that we treated with bendamustine alone had really pretty modest response to this treatment. Here are the survival curves, and I just show on the right, kind of the picture tells 1,000 words. So on the left, before treatment, you see all of these dark areas. This is a PET scan showing all of the areas of involvement. This is bladder, but the rest of this is all tumor. And you can see after therapy, almost all of this is gone. This is stomach, which takes some of the uptake as well. But all of the rest of this disease is gone now. There's no active disease. And you can see the survival curves they show at the top now, with roughly 40% of these patients durably disease-free and alive 3 years out from the treatment now. Again, a group of individuals that probably had a survival that would be measured on the order of months, if not maybe a year. So phenomenal success for our first product that we generated. And this was, I think, so exciting to the individuals who saw this and to the FDA that they granted us RMAT, or Regenerative Medicine Advanced Therapy designation, to treat patients with Hodgkin's lymphoma with this therapy, which, as I said before, puts us on the fast track to the clinic and allows us to do a more modest phase 3 study to show efficacy. So I've talked about kind of all of the really nice stuff about CAR T cells, and they're phenomenally successful when they work. They also have kind of a Dr. Jekyll, Mr. Hyde side to them. And the Mr. Hyde side to them is that they basically can get out of hand and cause significant complications and toxicity, including death in a handful of the first patients that were treated. And you can see kind of all of this comments that came out of studies that were done by company Juno, where initially 3 individuals and then 2 additional individuals died on their CAR CD19 therapy. And they mostly died of central nervous system toxicity. They developed cerebral edema from the proteins that are produced by these CAR T cells that migrate into the brain and can cause real problems. There have been deaths in almost every study of CAR T cells that have been performed, except at UNC. We have not had any deaths in the first 110 patients that we've treated. And part of that is that Gianpietro Dotti, 15 years ago at Baylor, realized that this was going to be a problem and engineered a safety switch that we can turn on and off to get rid of these CAR cells. And this is basically the safety switch that we put into the CARs. So we put in 2 different proteins. One is this inducible caspase-9 homodimer that has a drug binding domain on the end of it. This is an inert entity that doesn't do anything unless you add the drug. And when you add the drug, and the drug doesn't have any activity other than it will cause these 2 proteins to come together. When you add the drug, these 2 proteins come together within the CAR T cell, and they will activate a protein called caspase-9 that will kill the CAR T cell. And this occurs unbelievably quickly, within hours of giving this inert drug that all it does is activate caspase-9 to kill the CAR T cells. And so we treated a patient with refractory B-cell ALL. And again, approval for commercial drugs is for pediatrics and up to age 25. You can't treat these individuals above age 25. So this was one of the only avenues that this individual had. He received our CAR T cells that target CD19 with the inducible Cas9 safety switch. And then within a week of treatment, she developed severe complications with initially high fevers, low blood pressure, followed by severe neurotoxicity with non-convulsive status epilepticus. So she had multiple seizures, and then stupor where she wouldn't wake up. That lasted for 72 hours, despite standard of care treatment. And here you can see here, this is the dimerizing drug. So this black line is the CAR T cells. You can see the number of these rapidly going up. And then we've given all of the standard therapies back here, and nothing is causing these cells to come down. We give the dimerizing drug, and then within hours, the number of CAR T cells plummets from its high into the 14, 1500 down closer to 100. And then she goes from grade 3 to 4 complications to grade 1 to out of the hospital. So we've now treated 3 individuals with this approach, all of whom have done phenomenally well. And moreover, all of whom went into remission from their disease. So we have enough CAR T cells left after the therapy that the disease goes away, but we're able to ameliorate the complications that are associated with this treatment. And this is the publication that we had around the first patient in blood this past year. And then, we had a number of questions around solid tumors, and I'll talk about our pipeline in a second. This is work out of my laboratory. So Gianpietro Dotti and my group have focused extensively on trying to treat solid tumors, which are much harder to treat with CAR T cells. And that's predominantly around the fact that T cells normally migrate into lymph nodes and into the bone marrow where leukemia and lymphoma lives, but they don't normally migrate into the breast or the lung or the prostate or the colon, where most of the solid tumors that cause most death in the United States. And what we've done here is we've developed a new CAR T cell with a potent adjuvant that we can either get the CAR to generate or we can generate ourselves. And now we've treated mice with breast cancer that are unbelievably difficult. None of the other therapies that we've been able to generate can cure any of these mice. And I show here, this is kind of tumor growth in these mice. And you can see this is kind of conventional CAR T cells really don't work. These are the new CAR T cells with this new adjuvant either encoded in the CAR or given separately. Here you see kind of the tumor growth curves with the other CARs not working. And you see the negative effect of this CAR. And then you see the survival out to 100 days with now 75% of the mice now surviving without breast cancer. So we're in the process of developing this therapy for different types of breast cancer. This is for triple negative breast cancer. Gianpietro Dotti's got another target, B7H3, that he's also developing for breast cancer. We hope to be treating individuals in probably the next 12 to 24 months with that disease with these therapies. And then last, I just showed the pipeline that we have and then the funding that we have. We've been phenomenally successful in getting funding from both the National Institutes of Health. You can see we have 3 different NCI-sponsored studies. Stand Up to Cancer, we have the only Stand Up to Cancer grant in the state of North Carolina for our T-cell lymphoma study. And then from industry, where we have the only study outside of the University of Pennsylvania for solid tumors that overexpress HER2 from Charisma. But you see now that we've opened a study in ovarian cancer. Paula Gehrig's the PI. We've treated the first individual with that treatment, which is given actually through a catheter into the peritoneum where ovarian cancer tends to metastasize. We have one of the few pediatric trials in the country that treats the most common solid tumor in children, which is neuroblastoma and now osteosarcoma. And then we're about to complete our multiple myeloma trial, and there are now commercial therapies that have just been approved in multiple myeloma. We're about to complete that trial as well. And then I think we're going to move into glioblastoma, brain cancer, head and neck cancer, and potentially lung cancer in the next 24 months. So I think, the future is, I think, extremely bright for individuals with these diseases. I think we're learning a lot more about why these cells work or don't work in solid tumors. And I think the new approaches are, I think, much more sound and rational in their development, and I think will really be game changers for these patients. So with that, I'll stop sharing and I'll take any questions.
Okay, John, in the solid tumors themselves, as you talk about it, talk a little bit more about the unique challenges of trying to get the therapy that you have into the solid tumor so it has an effect. And as an investigator, your kind of thought process about the different solid tumors, why you might pick one over the other.
Yeah, so I think solid tumors for us have had three challenges. The first is to find targets. CD19 is a phenomenally good target because it's on leukemia and lymphomas, and while it's on B cells, so we get rid of normal B cells, you can make up for the loss of B cells by giving intravenous immunoglobulin for some period of time. So one of the challenges has been finding phenomenal targets, and some of the decisions around which tumors to treat tend to be which tumors have the best targets. And for instance, which tumors express this B7H3 protein that we have a CAR for. And then once we even find a target, the second, probably even more challenging aspect of this is to get the cells into the site where the tumor grows and then to expand. And solid tumors are a much more inhospitable environment than liquid tumors. They develop a fairly nasty microenvironment that tends to block the ability of immune cells to work, which is why, unfortunately, cancer progresses. And so we have to figure out ways to overcome that. And the second to last slide that I showed is having the CAR T cells generate a protein that activates a pathway called STING that appears to drive CAR T cells into the tumor site and allows them to expand substantially. And it's those types of developments that we need for solid tumor CAR therapy to be effective.
Okay. So a couple of questions related to the cost of a therapy like this. Walk through people how that happens.
Yeah, so I can walk through our cost and I can walk through the commercial cost. So these are expensive. I don't want to belittle that. I mean, they're expensive because they're living patient-specific drugs. So each time we make a CAR T cell product, it's from your own cells.
Right.
It's specific to you. So there's not a way to manufacture these in bulk like you would for a pill or a small molecule IV therapy. It costs us roughly about $25,000 to $34,000 to manufacture these cells per patient, and that's mostly around the cost to make the virus, and then the cost to grow the cells, and then kind of the maintenance cost to maintain the facility. The commercial cost for these is a log factor greater than that. So the commercial cost for Kymriah or Yescarta, which were the first two, are $405,000 and $373,000. And then the most recent treatments that have come from Juno and from BMS are in the same ballpark. So typically they've been pricing these close to $400,000 per treatment.
Okay. So one of the things that you talked about in describing the toxicity of it is using the protein that you induced to stop the CAR T cells to decrease the number, but you still had response. How does that help you plan for future studies about how many you might give a person? How does that help, or how does that inform you?
Yeah, so I think that the positive part of that for us is twofold. One is, as I said, the targets for solid tumors are a little more challenging to find in terms of specificity. You don't really want to look at targets that are expressed at a reasonable level on normal cells, because these cells can cause a lot of damage to tissues. But by having a mechanism to turn the cells off, we feel more comfortable about pursuing targets that I think other folks are more reticent to pursue, because we can get rid of any untoward effects if we see that. The second is, I think we can now extend the range of individuals who get treated. I mean, unfortunately, I showed a 26-year-old, and it's phenomenally successful, this therapy in young adults and children, and that's great. But the majority of people who get cancer are my age or older. And as you start to get into the 7th, 8th, 9th decade, it's really challenging to get complicated toxic therapy. And so one of the benefits of this approach is we can now take individuals in their 70s or 80s, and if they're frail, still treat them with confidence that if we have complications from the cells, get rid of them. So I think the benefit has kind of been on both sides for us. And for instance, the the GD2 treatment that we have for neuroblastoma incorporates the same inducible caspase-9 for children with neuroblastoma. And so if we see any complications from that treatment, we'll just get rid of the cells and move on.
Okay. So you talked about the neural complications. What are the other complications that you'll see of these types of therapies? Is it specific for that disease and the CAR T, or is it generically CAR T?
There's a fair number of generic CAR T cell symptoms, and they tend to be what looks to us like the flu on steroids. So it tends to be unbelievably high fevers, rigors, low blood pressure, and then leakiness of vessels that can lead to circulatory collapse and pulmonary complications. And then there's another process that activates macrophages that can lead to substantial liver toxicity. And so these have been fatal in individuals that have these. And to kind of bring it back to what you started with, they're not that hugely dissimilar from kind of the dysregulated immune system in people who get COVID-19 infection, who have more complications sometimes from the immune response than they do from the virus. This is kind of a similar process. And so it's treated similarly. So the treatments for COVID-19 are steroids and maybe tocilizumab. Our treatments are reversed. So this anti-IL-6 receptor drug, tocilizumab and steroids. But in the central nervous system, they don't work very well. And that's where this, I think, safety switch has been a game changer, because there really isn't any substantial therapy outside of high-dose steroids for those individuals.
Okay. So some of the people on today will have seen, heard, read about the CAR T therapy at other places like Penn, Baylor, you described others. What's the uniqueness of what y'all are doing here in comparison to those others?
Yeah, so I mean, so I think initially in the field, there were National Cancer Institute and then 3 centers that kind of were at the forefront of the field. One was Penn, one was Sloan Kettering, and one was the Hutch. And City of Hope had a modest sized program. And then there were a bunch of people who tried to catch up in the last decade, and the only group that's done that is us. So I think we've now moved into the forefront of that group. I think we're listed now as kind of one of the top 5 accruing and top 5 developing CAR T-cell places in the country, and one of kind of the cutting edge preeminent places in the world. And so all of the therapy that I talked about today, you can't get anywhere outside of UNC or Baylor, which is our partner in developing some of this. So if you wanted to get treated for ovarian cancer with CAR T cells, you can't get that therapy other than here. If you wanted to get the stand-up cancer T-cell lymphoma therapy, it has to be here. The neuroblastoma therapy has to be here. We're working with partners around the country to export our technology, Wesley. So it would be helpful, I think, for children, for instance, if you could live in Atlanta, get treated at Emory and stay there. But it's our therapy. It's not available anywhere but through us.
Okay. So, and you look at the people that have the extreme side effects that you've either, for y'all, you've had to give the drug that it will kill the cells. Others, they've had worse outcomes. Is there a type of patient, a phenotype that they're more likely to experience side effects, or there's a certain randomness to it?
I think there's a bit of randomness. There clearly are individuals who tend to do worse. So individuals with a lot of cancer at the time of their therapy tend to have more side effects. Individuals in some instances where we give a large number of the cells, tend to have a lot of side effects. But in general, there tends to be probably more randomness than we would like. And it's not clear always who's going to do well and who's not. Other than I think it's pretty clear that if you treat people with really aggressive tumors that are fairly prevalent at the time of therapy, they tend to have the most side effects.
Okay.
They tend to get the most benefit. So, I mean, it's a trade-off that we're willing to deal with. But, those are the people that we want to try to see if we can cure.
Okay. In some of the treatment algorithm you talked about use of drugs and then treatment. There's several questions related to monoclonal antibodies and people on monoclonal antibodies. What role does that play in CAR T?
It's probably not hugely specific to CAR T cell therapy, although we're clearly kind of the kissing cousin of monoclonal antibodies. So all of the single chain variable fragments that we generate, the things in the CAR T cell that bind to the tumor, come from an antibody. What we basically do is we either develop our own antibodies or we mine the literature for antibodies that have been generated against tumor proteins. And then we generate a part of the antibody, just the binding part, to put in the CAR T cell. So the CAR T cells that we have come from antibodies that were generated against CD19 or against HER2/neu or B7H3 or GD2. And for instance, the GD2 antibody is actually available to treat children. So this is an offshoot of that. It's different. And again, the upside to the CAR T cells is antibodies are phenomenally good at binding, but then they have to bring in parts of the immune system to kill the tumor cell. We don't need that. Once the CAR T cell binds to the tumor cell, it can kill itself. So it's clearly more effective therapy than the antibody, but they're offshoots of antibody technology. And then there are, I won't go hugely into this, but there are other things that people have developed that kind of mimic what we do with CAR T cells. And so what people have realized is maybe a good way to treat something with an antibody is make what's called a bispecific antibody or a direct antibody engager. And so what you do with these is you make an antibody that has both a part that binds to the tumor and a part that binds to a T cell. And so when the antibody binds the tumor, it brings the T cell in to kill it. And that's fairly similar to the approach that we have with CAR T cells, except we have it all in one package and they have to bring them together to do their work.
Okay, so the number of studies that you talked about, someone would fail first, second, third line treatment, and then you use CAR T therapy. In your mind, as you think about the future, does that ever change? Does it become frontline therapy, first line therapy for certain diseases?
Yeah, so I think that there's a large study being done right now nationally that's trying to figure out if CAR T cells targeting CD19 can replace stem cell transplant as a effective but less toxic therapy. And that data is hopefully going to come in the next year. That's, I think, where people would start to move this up into is get your initial therapy. If you're cured, that's great. If you don't, get treated again and then go through CAR T cell therapy and get around the side effects of stem cell transplant, which, as I talked about when I was in Seattle, graft versus host disease and all the complications of that aren't an issue with CAR T cell treatment.
Okay. So if you go out for some of the individuals you talk about, you're a good complete remission of 2, 3, 4 years, but then some have a recurrence at that point. Any thoughts about why that is, what you're looking at, what you're thinking about, why someone 2 years out would have complications at that point?
Yeah, so, and I think it's CAR specific. So we've seen in the CARs that target CD19, a lot of the relapses occur because individuals either lose expression of CD19. So if you lose the target on the tumor cell, then the CAR can't work, or they lose the expression of the part of CD19 that the antibody binds to. So the rest of CD19 is there, but they just lose the part that the antibody binds to. And then again, if the part of the antibody that's in the CAR doesn't bind to the tumor cell, it doesn't work. So that's probably 2/3 of the CD19 progressions. In the CD30 progressions, we've not actually seen CD30 loss. What we've typically seen is two things happen. One is we get diminished numbers of CAR T cells over time. And so there aren't enough CAR T cells to effectively survey for tumor cells, or the CAR T cells themselves wear out. And so there's a process by which T cells kind of lose function over time. That's how pembrolizumab and nivolumab, Opdivo, and Keytruda work. And we see that in our CAR cells. So they tend to basically tire out over time. And what we found, interestingly enough, in a handful of folks, this is a study done by a really talented fellow at UNC, Tim Voorhees, is he noticed that individuals who failed PD-1 therapy for Hodgkin's disease before CAR treatment, when they got the same treatment after the CAR, almost always went into remission and stayed in remission. And so we have a lot of active work now trying to figure out why would a treatment that didn't work beforehand work after you give CAR cells, even when the person relapses after the CARs. And he, was able to generate a substantial amount of data and grant funding around that. And that's an ongoing study that people can participate in if they're interested at UNC.
Okay, so a couple of really good questions related to kind of the mechanism of what you're talking about. The first one, you talked about the use of an adjuvant earlier. And so just a little bit more about generically, what is an adjuvant? And then how does it work? How does it help you in that particular description of what you described?
Yeah, so adjuvants are kind of the immunologist kind of dirty little secret about how you kind of generate an immune response. So, I think most people think of a vaccine as basically you find like the spike protein of COVID and you generate a way to produce that and put it into a person and you generate an immune response. But oftentimes we've got to couple that with some other inert substance that will activate the immune system, and that's an adjuvant. For the COVID vaccines, it's the nanolipid particles that do it. For the chimpanzee, for the J&J vaccine, it's the adenoviral thing that'll do it. And for what we do, it's this small dinucleotide that activates this sensor in the cells called STING. And so if we basically either activate STING ourselves or figure out a way to give the small dinucleotide, we now massively enhance the ability of the immune system to pull CAR T cells into the tumor where they normally don't do that. And so I think the most exciting part is the CAR T cells, where we basically worked out using that same prodrug that we use to kill the cells, we can use that to dimerize sting and activate the cells to markedly expand and increase their function in the tumor. But typically you have to do that. Or the other thing that tends to work as an adjuvant, oddly enough, is chemotherapy, which induces cell death, and cell death tends to activate the immune system. So there are some current studies where they basically combine chemotherapy with CAR T cells to try to see if you can get better expansion in solid tumors.
Okay, so another question, again, thinking about the mechanism of this, the question is about if you have a tumor with no identified targetable protein, what else are you thinking about that you're trying to target? And this question talks about non-genomic RNA, but what else, what are you thinking that you can target if you don't have that as an option?
Yeah, so I think there's a couple of approaches that folks have looked at. So we always have something on the surface of the cell. So there's always something there. The issue for us tends to be specificity. So is it something that's enriched on the tumor compared to normal? If it is, then we can target that. If it's not, one of the things that folks have done, including our group and Gianpietro Dodi's group, is make CARs specific for multiple proteins. So you have on-off switches. So you basically say, before the CAR can function, it has to have protein A on the tumor, protein B on the tumor, and protein C on the tumor. And when you look at it, none of the normal cells express that constellation of proteins. So there are ways to use and/or Boolean logic gates to turn on and off CAR T cells. And we've done that. Wendell Lim has been the person who's, I think, pushed that field the furthest. But both Pietro and myself have entities that work that in that way. The other thing that kind of we've been looking at is, in addition to CAR T cells, we can make T cells. So T cells recognize a entity that's composed of an MHC protein and a ligand, and the ligand is a peptide that's found from within the cell. The T cell receptor is not the best of all receptors in terms of killing. We can actually substantially enhance the function of a T cell receptor by taking it out of the body, working on it, and then putting it back in with a virus. And then we can make those T cells specific for a peptide from within the cell, rather than having to deal with things on the surface. So we've also got that. I think Barbara Savolda has been the person that's pushed that the furthest. We've got 3 grants around that area with Paul Armistead working pretty diligently to try to develop that therapy for acute leukemia that's going to be specific for a peptide that's found within the granules of the leukemic cells. So that doesn't require outside expression. It can take things that are just inside the cell, as long as they tend to be tumor specific.
Okay. So another question related kind of the mechanism of what we've been talking about. There's each person gets a certain dose of the CAR Ts. How do you decide on that? It's just the number of cells that you got through the process. Is there a certain number of cells you're giving everybody? How do you decide to give me a dose?
Yeah, so some of it's kind of done based on some preclinical work to see if there's a dose effect in animal models. Some of that's based on our prior work in other diseases that are similar to what we're treating, knowing what the effective dose is. And then some of that tends to be based in these phase 1 studies on where you land in the cohorts. Typically, for safety reasons, we start at the lowest dose and kind of work our way up. And we don't see much in the way of dose limiting toxicity. So as we expand the number of cells that we give, we don't see a lot of substantial increased complications. So usually we're limited by the number of cells that we can grow to what we can give, although there's really not a lot of data that going above a certain number of CAR T cells makes a difference. Okay.
So there's another question, and again, because of the news and the COVID vaccine, a couple of them being mRNA vaccines, how does, or what role does mRNA play in any of the processes that you're talking about?
Yeah, so the ovarian cancer CAR that we have is an mRNA CAR. So again, mRNA basically is the template for making proteins. If you put mRNA into a cell, the cell will recognize the mRNA and make the protein from the mRNA. In the case of the COVID vaccine, that's the spike protein. So you'll make the spike protein, which then can be presented and immune cells can recognize it and react against it. For the CAR that we make, we put mRNA for the B7H3 CAR. So the whole construct that the virus normally generates, we just have on an mRNA. The upside to that is there's no integration event in the cell, so that the mRNA doesn't persist. So it'll make the CAR once, And then as the cell divides, the CAR will be diluted out. So you don't have to worry about persistence of the CARs and toxicity. The downside is we have to give these every week for a couple of weeks, 3 to 4 treatments to make sure that they work. But it's the exact same technology that is used in the Moderna and the Pfizer vaccines. It's just encoding a different mRNA, in this case, the CAR.
Okay. John, there's so much information that you generate out of each patient that would just in a thoughtful way for each of those, like spawn a number of new studies. And then you've got all the different diseases, solid tumors versus non-solid tumors, different parts of the body. Just help the people that are on today. How do you personally, and how does your group, and how does the larger group at Lineberger, how do you walk through, like, because you have to focus and you have to pick out specific targets you want to go after. How do you go from a really broad range to pick those out? What's that process like for y'all?
Yeah, and that's a phenomenal question. And I think probably still an iterative kind of ongoing process. I mean, I think the first kind of principle that we look at in the group is what are the needs of the folks in the state of North Carolina? So what are the diseases that have increased prevalence in citizens of the state of North Carolina? And are any of those diseases ones that are difficult to treat? And so our initial foray into multiple myeloma came about because that's a disease that's overrepresented in individuals in the state of North Carolina and clearly overrepresented in Black individuals in the state of North Carolina. So we wanted to try to develop a therapy that could potentially treat that. The second is to try to look at, what are the diseases that are the most prevalent that cause death in the U.S. And so I think the proof of principle around hematologic malignancies is great, but I think more and more we've focused on kind of the big 4 or 5 cancers that tend to cause most of the death and trying to come up with ways to make those diseases, especially in the metastatic setting, less lethal. I mean, in almost all instances, metastatic solid tumors are unfortunately a death sentence for people who get those. And then, we work diligently in preclinical models with partners outside and inside UNC to look at antibodies, help to develop targets, help to develop other approaches. And then we have a phenomenal group of bioinformaticians that kind of crunch data for us and can hand us back information. And that's kind of where the PD-1 data came back, where they handed us back information that showed that, yeah, we have a lot of PD-1 expression on our CAR cells. And interestingly enough, when you got the anti-PD-1 therapy, now you responded again. So, the group of us, there's, I think, probably 70 of us over here that are working. There's 50 of us that do the clinical trials. I'd say it takes a village, but it maybe takes a small city to do this, Leslie. And so, we continue to learn from our colleagues and continue to try to push the field ourselves. So again, an exciting time, but those are real challenges for us. And, we spend kind of every day trying to figure out where we want to go.
Okay, no, it's unbelievable work, John. Leslie has something she'd like to show us, and then we'll come back for a final part.
All right, thank you so much, Dr. Burks. I'd like to just transition real quickly to show you all a video on our CAR T program and how it's affecting a particular patient.
Originally, they had me do just a strict chemotherapy regimen. And then they said, let's do a stem cell transplant. I couldn't eat for like a week. Major weight loss, hair loss. And then when it came back in 2014, they sent me to get something called proton therapy radiation. My whole face was a different shade, a total different shade, just because of the intensity of the radiation. It definitely took a while for me to get my strength back up after that.
Acute lymphoblastic leukemia, or ALL, is a bone marrow cancer. And in Sabrina's case, you've seen that over the last 11 years, she's had long treatments and quite a long story. Each one of those treatments has taken its toll, and we would love to be able to impact the cancer without impacting the patient quite so much. The CAR T-cell immunotherapy program is used to modify healthy cells in the immune system of these patients to attack the cancer cells with minimal toxicity. In contrast to a drug, which is given in several phases, one advantage of CAR T cells is that they can survive and persist within the body and be an occupying force, if you will.
My oncologist at UVA said, let's send you to UNC to have CAR T cell therapy, because I think this clinical trial would be the best option for you, and it would be less taxing on you than any other treatment that you've had. I was still skeptical, but I never recovered from a treatment as fast as I recovered with the CAR T cells.
So at UNC Lineberger, we're fortunate enough to be one of the few centers in the country to have a pharmaceutical grade facility, a great team of researchers and clinicians who are designing these therapies, making sure the therapies are safe, and then bringing them from the laboratories to the bedside and delivering them to patients. By having such a coherent team, we can speed that discovery and speed the development of therapies and get them back to patients to let them get back to their lives quicker.
These past 5 months since CAR T, I feel way better than I had imagined in my mind. Now I'm gonna get out there as much as I possibly can. I'm not gonna let cancer define me.
John, I've seen that video innumerable times and I still get emotional every time I watch it. And so I can't imagine for you and all the patients that you are working with, how rewarding that is for them and you. Just to end the hour, if we think about another 10 years, what do you need from us to expand, develop your program into the place that you would hope and dream about?
Yeah, and thanks, Leslie, for that. I mean, we love to see Sabrina every time that she comes here. It's a phenomenal story, and she's a phenomenal woman. I kind of hate to say it, Wesley, but resources. We compete with groups that have substantial backing from pharma companies. Again, I think that we can compete intellectually with anybody in the world for what we do. But in order for us to kind of expand our scope and reach, resources, the GMP, which you saw as the manufacturing place for Sabrina cells, is extremely expensive to maintain. It's hard to get grant funding for that because it's just a manufacturing facility, basically a pharma company. So resources around that, and then just everybody who's involved. You saw Matt Foster, the clinicians. Catherine Chang was mentioned. She's our main research coordinator, and then all of the folks over here who are doing the work. So. I think we've made phenomenal strides, and we've been phenomenally resourced by the Cancer Center and by the state of North Carolina. But I think for us to get to the next level is going to take a next level jump in resources that I think we're anxious to try to get there.
Okay, John, thank you again for just taking the time to talk about the work you're doing, the passion you have for it, and your hopes and dreams for the future. So I'll turn it back over to Leslie.
Thank you, Dr. Burks and Dr. Serody, for just a really wonderful and fascinating presentation on our CAR T program. If you all are moved by what you've heard today and you want to support the work of Dr. Serody and his team, I hope you will consider getting in touch with us and or making a donation to our clinical immunotherapy research fund. That link is actually in the chat there. We'd love to continue the conversation with you. I also wanted to say that we're going to continue the conversation on cancer. We'll take a little break during the summer with our Big Game Changers series and pick back up in September with more conversation around the cutting edge research, patient care, and education that's happening at UNC Lineberger and the North Carolina Cancer Hospital.